Hello everyone. Thank you for joining me today for this webinar on advancing immunotherapeutics research with immuno-oncology with Luminex technology and MILLIPLEX multiplexing assays. My name is Amy Johnson and I am the immunoassay platform specialist in the Mid-Atlantic. So if your lab is located in Virginia or Maryland, I'm your immunoassay specialist. My contact information is on this slide and I'll also be presenting it again at the end of my talk today. So during today's session, we're going to cover MilliporeSigma's immunoassay portfolio and the different types of assays that we offer. And then we're going to focus in on Luminex xMAP technology and those instruments that are available, and our MILLIPLEX protein multiplexing assays that are read on Luminex instruments. We're going to highlight specific panels, that MILLIPLEX panels that have been developed with immunotherapeutic research in mind. We're going to summarize what we discussed today and close everything out. So let's start with talking about MilliporeSigma's immunoassay portfolio. So as I said, I'm part of the dedicated team of immunoassay specialists and immunoassay field application scientists that cover North America and really the world. Our portfolio is one where we follow a philosophy of fit for purpose. So what does that mean? That means that we offer a variety of products meant to fit specifically your needs for your research. Not too much, not too little, just the right fit for you. So if we start all the way on the left of the slide with our ELISA portfolio, now that EMD Millipore and Sigma Aldrich are combined into a single company, our ELISA portfolio approaches near 2,000 different ELISAs. So ELISAs are the gold standard protein quantitation assays. It's technology that's been around since the 70s, and it's tried and true and probably very familiar to many of you on this webinar today. So what do you need to run an ELISA? You need a plate reader, whether it's colorimetric or fluorometric. And with ELISAs, you are quantitating a single protein at a time. The sensitivity range of ELISAs tends to be about in the picogram per milliliter range, and you need about 50 to 100 microliters of your sample, depending on the analyte that you're quantitating. We're looking at a dynamic range of about 2 to 3 logs with a traditional ELISA. If we go all the way to the right-hand side of the slide, you'll see that purple box. That's our SMC X Pro. SMC stands for single molecule counting, and the SMC X Pro platform is our ultra-sensitivity or ultra-sensitive immunoassay platform. So with these assays, we're again looking at a single protein to analyze and to quantitate. But the power here is that the sensitivity levels of this platform reach down into the femtogram per milliliter range. So again, depending on the analyte being quantitated, this could be 100 to 1,000 times more sensitive than those traditional ELISAs we just talked about. So this platform is really ideally suited for very low abundant proteins, or perhaps if you're conducting a study where the endogenous levels of the protein or your biomarker of interest are very low, but then in your diseased population or your therapeutic intervention population, that same biomarker may be found at very high levels. Because of the very wide dynamic range of the SMC platform, you'd be able to capture and quantitate that protein in a single assay from both of those populations. This allows you to not only reduce variability in the data you're collecting, but possibly also stratify between those groups. There may be instances where the changes between your control and your patient population are very small, but very significant changes. And again, because of the dynamic range and the ability of this platform to resolve between those two groups, you can capture all of that data that you need. But today we're going to focus on what you see in the middle of the slide, Luminex instruments and our MilliPlex protein multiplexing assays. So Luminex technology, as you'll see in a few slides, is built, it's an ELISA-based system, but these ELISAs are built on the bead. Because of this, we can simultaneously quantitate many proteins in a single sample all at the same time. So this is really, this Luminex technology is ideal for higher throughput screens, or perhaps if you're working with a new model system and you don't really know what to expect. So if you're doing screening to figure out what your targets, what biomarkers are changing in the most relevant ways in your new model system. Like traditional ELISAs, the typical sensitivity range of a MILLIPLEX assay for a Luminex instrument is about a picogram per milliliter. If you're working with serum and plasma, you would need no more than 25 microliters of your sample per well. We recommend that you run your assays in duplicate. So if you think about just that enhanced throughput, you're going to see that our largest human MILLIPLEX assay that can be run on the Luminex instrument, it's an off-the-shelf assay and it quantitates 48 different proteins. It's our human cytokine chemokine growth factor panel A. So in just 25 microliters of sample, you are quantitating 48 different proteins. The dynamic range of these assays tends to be about 3 to 4 logs, so about the mid-range between an ELISA and the SMC X Pro platform. So let's shift a little bit and look specifically at Luminex technology. Luminex's technology is xMAP technology. So Luminex is a standalone company. They are based in Austin, Texas, and they developed this ELISA on a bead technology that I'm going to describe for you next. So the xMAP beads that are used for these assays are made and hard dyed with a combination of a red dye and an infrared dye. Different ratios of those dyes in the beads allows us to construct a spectrum of these bead colors or bead regions or spectral address. These are all terms that you'll hear used interchangeably with these types of assays. So on one end of that spectrum, we may have a bead color that is all red dye and no infrared dye. On the other end of the spectrum, we'll have a bead color that's all infrared and no red, and then an array of colors in between. The Luminex instruments that you see on the bottom of the screen, the different models are designed to be able to classify or differentiate between those bead colors. Because of that, we can mix beads of different colors together in the same well. Those different colored beads are coated with different capture antibodies against a different specific targets. So that's how we can build these multiplexing assays where you can simultaneously quantitate so many different proteins at a time. There are a variety of Luminex instruments available, starting with the MagPix all the way on the left that you may see in some labs, the LX200, the FlexMap 3D, and the newer, brand new, just launched xMAP IntelliFlex instrument. So if we look at the MILLIPLEX assays that MilliporeSigma builds to be read on Luminex instruments and how these assays are actually working. So we already discussed that these MILLIPLEX assays are based on the xMAP technology, and you're simultaneously measuring multiple analytes in a single well. Typically 1 up to 40 plus different analytes in a single well. You can use these MILLIPLEX assays to quantitate soluble cytokines, chemokines, growth factors that are being produced and released either into serum and plasma or cell culture media, perhaps. We also offer intracellular signaling MILLIPLEX assays, and we'll discuss these toward the end of the webinar as well. These I want you to think about more like Western blots, where you're using cell or tissue lysates, and you're looking at perhaps phosphorylated versus total amounts of a protein in a signaling pathway like AKT or mTOR. For all MILLIPLEX assays, if you're working with serum or plasma, cell culture supernatant, again, you're looking at needing about 25 microliters per well of sample in order to run the assay. You may also use tissue lysates, cell culture lysates, and we have modified protocols for smaller sample volumes if that's what's available, and also many different sample types. So if we look about how these MILLIPLEX assays actually work, the cartoon at the bottom is going to take you through step by step the way most MILLIPLEX assays work. You're going to see that they're a sandwich ELISA format. So if we have that Luminex bead that I described to you that's hard dyed with infrared and red dye, and that is coated with a capture antibody. So for example, if we wanted to quantitate the levels of IL-6 in a sample, this capture antibody would be an anti-IL-6 antibody. So we have these beads already coated, and you incubate them with your sample. This allows for interaction between the analyte in your sample and that capture antibody. We then go on to build that sandwich. So the next step is to incubate that with a detection antibody, or detection antibody cocktail, actually, in terms of MILLIPLEX assays. The detection antibodies are biotinylated, and we use that because the phycoerythrin, or PE, that is the fluorescent reporter molecule for all of these assays, has a streptavidin conjugated to it. So we're using that biotin streptavidin interaction in order to attach the phycoerythrin to complete our sandwich. The plate is then put in the Luminex instrument for reading, and depending on the type of instrument, the bead color is excited either by an LED, a red LED or a red laser to tell the bead color. The instrument sees that fluorescence and classifies the bead accordingly. And then a green laser or green LED excites the phycoerythrin. And the median fluorescence intensity from that phycoerythrin reporter is the raw data for all MILLIPLEX assays. We offer MILLIPLEX assays to cover a wide range of research areas or research topics. So inflammation or classic immunology type assays looking at cytokines, chemokines, growth factors are widely available. We also have panels specifically built to look for neuroscience targets, A-betas, taus, and so on and so forth. We have panels geared toward assessing metabolic status and cardiovascular diseases, diabetes, and obesity-associated biomarkers. We have many panels that look at cancer biomarkers, circulating cancer biomarkers, as well as intracellular. Other infectious disease states, drug safety and toxicity, particularly in terms of kidney damage and liver damage. And also we have people that are doing pharmacokinetics and pharmacodynamics, more bioanalytical type assays using this platform as well. So when we think about the types of samples that you might be working with, I had already mentioned serum or plasma, cell culture, supernatants, but I don't want you to think that we are limited to those sample types by any means. So with MILLIPLEX assays, you're going to see protocols written out for serum and plasma, cell culture, supernatants, because they are the most common matrices that we have researchers working with. However, we have protocols worked out and recommendations for just about every sample type you could think of. Synovial fluid, cerebrospinal fluid, tissue homogenates from any tissue, nasal washes, tears, bronchial alveolar lavage fluid, urine, sputum, so on and so forth. So if you find yourself working with one of these more non-traditional sample types, please contact either our tech support team or your local immunoassay specialist or field application scientist, so they can give you recommendations on prepping these samples and amounts of these samples to use in your assays. So we're going to look a little bit more specifically at some of the immunotherapeutic geared MILLIPLEX assays that we have available. All the way on the left-hand side of the screen, you see the full panel for the human cytokine, chemokine, growth factor panel A, 48-plex MILLIPLEX assay. The list of proteins that you see listed there are all of the ones that are quantitated within that assay. In the middle of this slide, you see the panel, the Human Oncology Checkpoint Protein Panel 1. This is a 17-plex assay. And then we have a newly launched Immuno-Oncology Checkpoint Protein Panel 2 assay. This is a 31-plex. So the way the majority of MILLIPLEX assay kits work is that the full panel, so in terms of panel A, is the 48-plex. But it might be that you don't need to quantitate all 48 of those proteins listed there. The assays that you see listed here, and again, the majority of MILLIPLEX assays are what we call, what we term as are configurable. That means you can run a panel, you can run an assay that will quantitate all of the proteins you see listed, or you can pick and choose only the ones of interest, and we will build the assay accordingly. MILLIPLEX assays are priced according to the number of analytes or proteins that you're quantitating within that kit. So what my recommendations tend to be is if you're using a new model system, or it's a new study and you're not quite sure what to expect, for panel A, I would suggest running the full 48-plex once as a large screen to get an idea of what biomarkers are changing in meaningful ways in your model system. You may want to consider running it twice just to confirm, just to be sure. But then going forward, it might be that only 10 or 12 or 20 of those 48 proteins are really changing in meaningful ways. So going forward, we can build you assays to quantitate just those proteins. Because of the way that we develop our MILLIPLEX assays, the data that you get for a specific protein will not change depending on the plex size of the assay that you're running. So you might be thinking a 48-plex assay across how many different samples, run in duplicate, how am I possibly going to analyze all of this data? And you can generate a lot of data very quickly, which is of course the strength of Luminex technology and MILLIPLEX assays, but we also don't want it to bog you down and for you to spend too much time doing this, because you want to learn from that experiment and then move on and start planning your next steps. So we've developed our BELISA immunoassay curve fitting software to make this process very easy for you. So as you see, at the end of an assay, you can end up with a very large amount of data coming off of your run. What you would do if you had the BELISA software is that you will take your raw data file from your Luminex instrument, the .csv file, and you literally drag and drop it into the BELISA immunoassay software window. It will then plot your standard curves. It will show you your controls and your treated groups. It's also going to plot your QC controls. All MILLIPLEX assays come with high and low QC controls and the expected results from those QC controls. So that this has several purposes. One, if you do an assay run and your data looks a little strange, the first thing we want to do is look at the QCs and ensure that all the critical reagents in that MILLIPLEX assay kit perform to spec. Two, we work with research groups that conduct longitudinal studies perhaps over many, many years. The QC controls included offer a way for those researchers to monitor the performance of their assay from plate to plate and year to year throughout the course of your study. So what do you get with a MILLIPLEX multiplexing assay? The bottom line is you get everything you need except your samples that you want to analyze and some really clean water. Of course, we would always recommend MilliQ water for this. So you get the beads that are already conjugated with those capture antibodies. You get diluents, all buffers needed, assay and wash buffers. You get the full assay protocol booklet. You get the 96-well plate and the plate sealers needed to run the assay. You get your detection antibody cocktail, that streptavidin phycoerythrin reporter that I mentioned, those high and low QC controls. And we also include a serum matrix reagent. The serum matrix reagent is included and used by those of you who are using serum or matrix as your sample type. The reason it's included is that because in all immunoassays, we have to be aware of something called matrix effects. Matrix effects occur in immunoassays. They're hard to pinpoint, but they can affect your data in very real ways, and so you have to have an awareness about them. Basically, when you're running an immunoassay, if your standards are made up in a buffer like water or PBS even, something very clean and simple, and you're using that standard curve in the assay, and you're building that standard curve and calculating concentrations in your unknown samples, which if they're serum or plasma are very complex systems with many different proteins in there, you're not comparing apples to apples. The serum matrix component is included to add to your standards so that you're sure that the antibody binding kinetics of your standards are similar to the antibody binding kinetics in your unknown samples. If you do not include some sort of matrix control in your standards, what you end up doing is not properly shifting the standard curve to reflect the environment of your unknowns, so that your standard curve slope ends up being far steeper, and you end up undercalculating the amount of analyte or biomarker in your samples. So the serum matrix component comes standard in all off-the-shelf MILLIPLEX assays. If you're working with a sample type that is not serum or matrix, we have recommendations for what your matrix should be. So again, please reach out to tech support or your local specialist or immunoassay field application scientist. Okay. So the human panel A and the 2 immuno-oncology panels I introduced to you are typically being used by researchers for circulating biomarkers. But what if you wanted to look intracellularly? Maybe at some signaling pathways in cell lysates or tissue lysates. You want to see what pathways are being activated in your treatment or your engineering. So this introduces our cell signaling multiplexing assays. These again are similar in workflow to what I described to you earlier in terms of using coated beads for capture antibodies and building a sandwich. These panels are designed to look for a specific cell signaling pathway. So here we have AKT mTOR phosphoproteins, STAT phosphoproteins, MAPK phosphoprotein pathways, and RAS-RAF oncoprotein panel. When you're thinking about these assays, you can think of them more in terms of the Western blots that you might be doing. These are qualitative immunoassays. So the controls included are not standards. We're not building standard curves, but they're rather a yes-no sort of positive-negative dichotomous kind of readout. So the controls that you get for these assays are lysates that have been treated with different factors to either induce or inhibit activation of certain proteins. When you're doing a Western blot, you have to be careful about loading equal amounts of protein in the wells in your gels. The same case is here, and there's a couple different ways that you can go about normalizing loading in these assays. You could either do a protein assay and know that you're loading the same amount of protein, typically somewhere between 1 and 40 micrograms of protein per well, total protein per well. We also offer loading control beads that can be added into these cell signaling panels. So for beta tubulin or GAP-DH, the readouts from those beads could then be used as your normalizing loading control. To give you an idea of what this data typically looks like, we have a comparison of a Western blot versus a MILLIPLEX cell signaling pathway assay kit. So we're looking here at lysates from HeLa and HepG2 cells treated with different reagents, so insulin, wortmannin, rapamycin, LY, and so on and so forth, and looking for phosphorylation of AKT at serine 473, GSK3 beta at serine 9. So you see those dark bands, that is the Western blot data that probably looks familiar to a lot of you. And below with those pluses and minuses show the MFI data, the median fluorescence intensity data, that is the fluorescence data from the MILLIPLEX assay and the Luminex instrument reading. So you can see that they're very highly correlated to each other. These assays are built to give you reliability in your results. So in terms of concentrations or degrees of change, if we look at the Western blot in the middle and then what the corresponding MFI from the MILLIPLEX assay looks like, you can see very high R-squared values indicating high correlation between the two. Just like with the soluble protein MILLIPLEX assays, the cell signaling assays, again, can't come with everything you need except some water and your samples. The coated beads, your detection antibody cocktail, the streptavidin, phycoerythrin, positive and negative controlled lysates, lysis buffer, assay buffer, and an amplification buffer that's used in these assays, your protocol booklet, your 96-well plate, and plate sealers. So to summarize what we've talked about today, we've talked about MilliporeSigma's immunoassay portfolio ranging from ELISAs through Luminex-based MILLIPLEX technology, multiplexing technology, all the way through our single molecule counting ultrasensitive immunoassay platform. In terms of MILLIPLEX assays, we are constantly developing new panels and actually very much welcome feedback from you all in terms of panels that would be of interest to you that we should develop. So please, anytime, we always welcome those ideas. This slide gives you an idea of the different types of assays that we have available and for the different species that we have available. So our largest portfolio is available for human proteins, quickly followed by mouse and rat. So, standard research models species. We also have non-human primate assays, and then assays for a variety of more veterinary models. So equine, feline, canine, bovine, porcine, ovine, and our very new chicken cytokine, chemokine panel. So MilliporeSigma's immunoassay specialist team offers the complete package, everything that you need in order to run successful immunoassays. Of course, we have the assays themselves, the instruments, whether it's a plate reader for your ELISAs, a Luminex instrument for your MilliPlex assays, or an SMC X Pro for your ultrasensitive assays. We have the Belisa analysis software to make your immunoassay data analysis as painless as possible. We do have custom assay development services. So if you don't find an assay on our website or our Analyte Quarterly that you need for your research, please reach out. It's possible that we could develop one for you as a custom assay. We have technical support, whether this is you calling into our technical support scientists, they are based in St. Louis, or working with myself or our field application scientists for on-site technical support in your lab. We want everyone to be successful. We are here to support you, so please don't hesitate to reach us, to contact us if we can help in any way. And with that, I'd like to thank you for your kind attention today. I really do appreciate you coming. Again, my name is Amy Johnson, and I'm on the immunoassay platform specialist team. I cover Virginia and Maryland, and my contact information is on this last slide. So please reach out if there's anything I can ever do to help. Thank you all, and I hope you enjoy the rest of the show.